All right, welcome back to the third part of our lecture series on the evolution of vertebrates. Today we're going to look at the evolution of fishes, specifically Chondrichthyes, so the cartilaginous fishes, as well as the bony fish and lobe-finned fish. Last video series, we talked about the evolution of the mechanics of the jaw, and as well as the agnathans, so the jawless fish, of how that transition from no jaws like the lamprey here and hagfish, not pictured, to our jaws that we're very familiar with, especially in the sharks and things. So jumping right into it, just to focus on the taxonomy again. The taxonomy typically gets a lot of students as it's just a lot of weird names. So you just have to spend some time with it. I'm going to reintroduce it here as well. So our last video series, we talked about the superclass Agnathans with the class Myxini, the hagfish, and the class Cephalospiniomorphii with the lampreys. Now we're moving on to the superclass, which we're probably more comfortable with. These three classes today: the Chondrichthyes, the sharks, the rays of chimeras, Actiniopterygii, the ray-finned fish, Sarcopterygii, or the lobe-finned fish. There's an Osteichthes, which is basically just classified as bony. We'll talk about those in our last video series. We're going to look at the evolution of tetrapods, so the class Amphibia, Reptilia, Aves, and Mammalia. So looking within the class, the extant fishes today, remember there's Cephalospiniomorphii, there's Myxini, so lampreys and hagfish, respectively, shown down here, hagfish and lamprey, and Chondrichthyes, and you can, some taxonomists will throw in bony fish, Osteichthes. So you got Chondrichthyes, which is, remember, chondra means, or chondric means cartilage, osteoporosis, if you're familiar with that, being of the bone. So you've seen this phylogenetic tree before, but again, it just paints a good picture of where we're at and where we're going. So again, last video, we talked about the agnathans, and we introduced the placoderms, so going from jawless to the extinct jawed lineage of the placoderms, and now we're continuing on in our evolutionary studies and looking at Chondrichthyes and Osteichthes, setting up the last video series again, being the tetrapods. So, in the class Chondrichthyes, that includes sharks, rays, and chimeras. Extremely successful class, over 750 species. Again, they're cartilaginous. Excuse me, I had a little allergy issues this morning. And they have paired appendages, very, very, very acute sentence, which we'll talk about and watch a video on. And part of their acute sentence comes from what's known as their lateral line. So remember, lateral, if it just goes left to right or horizontally. And so if you've ever caught a fish, you may not even notice this, but almost all fishes have, around here specifically, a lateral line. And this lateral line is running horizontally across their bodies and allows them to detect changes in water pressure. It's basically an extension of their ear and their nervous system. It's incredibly innate, incredibly tuned, and allows the fish, and especially the shark, which we'll watch a video in a second, to know exactly where an animal is in the water at all times. So it doesn't necessarily rely strictly on smell or strictly on eyesight. It has the ability to sense those changes in water pressure as an animal swims through the water. It's incredible. So fish exhibit decephalization. We've seen this vocabulary word before, but cephalization is just the orientation of neural or neuron structures, the brain, in the head region. So they have lots and lots of chemoreceptors. So let's break this word down here. Chemo being chemical. Obviously, a receptor is something that's receiving, which allows them for incredibly sense of taste and smell. So now we're actually starting to see some nervous structure differentiate into the ability to taste as well as to smell. The lateral line system, again, allows them to sense movement. And check this video out again. Yeah, check this video out of the shark's inner ear. This lateral, absolutely remarkable uh, adaptation that sharks and fishes have evolved to have. And so again, just underneath the skin there. And again, if you look at typical fish there, so you can see kind of right there, the changing color is usually what marks around that lateral line there. And so dorsal side, ventral side, and again, it's filled with fluid as a fish moves around or sometimes moves the water. These fish have the ability to sense it. Absolutely crazy. So looking again within the class Chondrichthyes of sharks, rays, and chimeras, lots of different diversity. Again, over 750 species of these guys here. Chondro is Greek word for cartilage there. Typically, sharks have over 3,000 teeth, which is just crazy to think about, and 6 to 20 rows, and they just have a steady supply of fish. So as a shark loses a tooth, another one's right there behind it, which is probably why you see, we have so many fossils of shark teeth because there's just so many of them. Not all of our sharks are carnivorous. Some of the biggest ones, like the nurse shark and whale shark, are filter feeders. Some have flat teeth for crushing mollusks. So again, think about kind of like your starfish there. These little flat mandibles there will help crush those mollusks. Some characteristics that we have in our hot sheets there. So again, these possess jaws. They do have some scales as well. And so when you dissect your shark, you're going to be able to sense and feel those scales are smooth going from head to tail. But if I go the opposite way, from tail to head, you're going to feel just how rough these scales actually are. 5 to 7 gills. They do have one heart there, and a lateral line. No swim bladder, which is kind of interesting to think about. So the function of a swim bladder is kind of like the ballast in these tanks and submarines. Is if I want to sink, I'm going to decrease my swim bladder so that I take on more water. And so, if the fish wants to go down toward the bottom of the lake or the ocean, it will decrease the swim bladder and it will become less buoyant. Sharks don't have that ability, and so what they've been able to do, which is just remarkable, and you'll sense this, you'll get this appreciation when you dissect the shark, is their liver. Their liver is very, very, very big. It's 3 lobes, and it just secretes a copious amounts of oil, fish oil. And so you already know from your macromolecule lessons and studies early in the year that oil and water don't mix, right? So oil is hydrophobic. And so, because of its hydrophobic principles, depending on how much oil or less oil I make, allows me to go up and down in the water. It's just absolutely crazy to think about. This heteroserial tail here, so the very stereotypical shark-looking tail or fish-looking tail. And this body plan has been tremendously successful, which kind of accounts for that there hasn't been much of a change, even in the fossil record, for long, many millions, hundreds of millions of years now. So our modern sharks, again, feeding type. Not all are carnivores. We have some parasites, believe it or not, and some are herbivores. Here's the basic shark anatomy that you'll want to know, familiar with before you dissect your shark. So again, find something that you can always find and then work a way around it. And so typically with the head region there, obviously a lot of names we're very familiar with. Nostrils, eyes, teeth. They do have a gill slit there, those 5 to 7 gill slits. Spherical there, we've seen this term in our arthropod study. We've got dorsal fin, we've got a second dorsal fin or caudal fin. So instead of saying tail fin, we say caudal. Anal fin, you can tell if it's male or female based on the if the fish or if the shark has claspers. And so these claspers are very bony fin on the ventral side. And that, as the name implies, is what's going to be used in reproduction, where it's going to clasp onto the help clasp onto the female genitalia and ejaculate the sperm. Kind of neat. Pelvic pectoral fins, pelvic fins are are sharks that only eat plankton and filter feeders. There again, these are usually big sharks, But look at the gill arches there. So just and this just incredible adaptation that they're able to open their mouth, creating this large vacuum where water goes from high concentration to low concentration. So when it closes its mouth, there's not a lot of water in its mouth. So as I open my big mouth, water is going to pour inside. Carnivorous, again, we're very familiar with the movie Jaws by this point. So we're very comfortable with carnivores. And there's even some parasites, like this cookie cutter shark. They're going to live inside other animals, which is just crazy to think about. And inside the womb, because sharks carry, not all, but many sharks carry their young inside them, excuse me, that baby sharks actually will eat their brothers and sisters. And so by the time they're actually born, these guys have eaten and killed dozens, if not hundreds, of individuals before they're born. So they're just absolutely already born to kill, which is crazy to think about. The Ampullae of Lorenzeni is a very unique adaptation that allows them to detect weak magnetic fields produced by other fish. So we all have a little bit of magnetic fields there generated from our action potentials firing. And it looks like little small whiskers or, like a 5 o'clock shadow. If you're a fan of the series The Flash, you may have heard or seen this when The Flash created this when King Shark was incapacitated momentarily. They generate electric field through his Ampullae of Lorenzeni to revive him. Kind of a neat, this complete nerd out moment for me. When I saw that, I was like, "Oh my God, look at this! This is crazy." So yeah, it's Hollywood and writers are fairly in tune with the biology of the world, so it's pretty neat. So the way this kind of works here, again, very similar to lateral line and the inner ear there, is that they have these little sensories on the nose area. And as these magnetic fields come in there, it's going to trigger these sensory cells, which then send nerve impulses to the brain to allow the fish to detect in front of him those electric signals, magnetic signals. So, as I mentioned earlier with development here, so there's some vocabulary for you. We got ovipary, ovivivipary, and vivipary. And so I've tried to condense and make these as good, just working definitions to help us out. As you guys know, I'm not very great with memorizing definitions, but I'm really good at examples. And so ovipary eggs are enclosed in a capsule, and they're laid outside their mother. So think of like a chicken, right? So they have the egg, the embryo is inside the egg is laid outside, chicken, right? A lot of your fish do the same thing. Ovivivipary, or what some of the sharks do, is they're actually going to give birth to live young, but they actually have their eggs inside, their fertilized eggs inside their uterus. So there's no placental connection. It's actually eggs, and so these eggs will hatch inside their mom. And again, that's why baby sharks will sit there, and the first one or couple is born have an advantage over the ones who aren't born yet, because the sharks will begin to eat the eggs in their younger brother and sisters, which is just crazy to think about. Vivipary is what we're very familiar with, is that it has that placental connection, and we're going to give birth to live young. So humans, for example, are a great example of that we're familiar with. So again, just a phylogenetic tree with the artistic representation of fish. So we're going to look here, and we're going to kind of go in this direction, left-hand side here. Then at the end of the lecture, we'll come back and look at the Sarcopterygii, the lungfish, which is going to give rise to the tetrapods. But so let's keep looking and keep going from the Chondrichthyes to the Actinopterygii to the Osteichthes, right? So the bony fish. So flatfish, eel, which is kind of weird thing about sturgeon, seahorses, all these make up the Osteichthes. So again, if you're following along on your hot sheet here, we've got possess jaws and teeth. It is a bony skeleton, very familiar with there. 4 paired gill arches, covered by something called an operculum. And the operculum is basically a bony gill flap that's going to protect those gills. And I'll show you a picture of that in just a second. Do have a swim bladder, a lateral line there. Most have scales, not all, but most have scales. Intestines, there is no spiral valve, so essentially no uh, long appendix-like structure there. Um, have paired fins, allow for the evolution of the tetrapods. That's really depending on what lineage and taxonomy you go to. So the Osteichthes encompasses both uh, Sarcopterygii and Actinioterygii. So this is kind of a different taxonomification. So for our sakes, we'll basically say that we're back here. So the osteo could kind of go right here. And so from the osteo kind of like being a superclass, if you will. And then we have these classes here, depending on the taxonomy that you follow. And so Actinioterygii are typically what we think about when we think of fish. Sarcopterygii, again, are going to be developed into later on in the tetrapods. Okay. So let's look at the structure of a bony fish. So again, you'll want to know your anatomy, external anatomy of the normal fish. And so we've got the operculum here, this little gill flap there. Typically, a lot of fish have two dorsal fins, a primary, which is usually spine, and then some type of ray. In catfish, this can be an adipose sometimes. Caudal fin, anal fin, pelvic, and pectoral fins. So lots of fins there to help them move around and swim in the ocean and in the freshwater fish. Here again, here's the operculum. So again, if you're familiar with noodling or hand fishing, what you'll do with catfish is you'll stick your hands inside their mouth, and then you're going to go through the mouth, and you're going to try to grasp the backside of the operculum before lifting out of the water. So decent rush if you've never done it. To go with somebody in your yard doing it because it is dangerous. So. But it is a good rush. Bony fish there, no surprise, their skeletons are made of calcium. Again, very familiar with these type of fish, especially in our Tennessee region. Trout, crappie, catfish, sunfish, and depending on where you brim, we're more comfortable calling them brim or panfish. Largemouth bass, maybe some smallmouth bass, but we're very familiar with these bony fishes. Some more vocabulary for you. We've got two different types or ways in which fish breed, right? So we're very familiar with the spawning aspect of fish. So essentially all fish kind of trying to mate around the same time. But we have two different types of ways, or two ways we could do that. One is androgynous, which is that as an adults, they're going to spend most of their life in the ocean. But when it's time to breed, they're going to make their way back into fresh water. So again, salmon, great example of that. So again, try to connect an example with this definition. Catadromous is the opposite, where they're going to spend most of their life in the fresh water, but they're going to move to the ocean to breed. Unfortunately, off the top of my head, I can't think of a good example of a catadromous fish for you there. So my apologies. So more body systems here. Feed them lots of different ways, parasites, herbivorous, carnivores. Most fish will use their gills to exchange gases there to breathe. So their gills are lots and lots and lots of surface area there, lots of feathery filaments to help increase the ability to exchange carbon dioxide and oxygen in the water. They have a two-chambered heart, and it is a closed circulation, a closed circulatory system, which gives them an advantage that we talked about with the mollusk or many mollusks in the open circulatory system. So I have a ventricle and an atrium, and so the atrium is going to pump blood to the gills, and the ventricles is going to pump blood throughout the body, just like in our hearts there. So fish digestive systems are very similar to humans. Excretory is a little different, as you can imagine. And so we're going to look here at the evolution of the kidney. We've kind of been looking at the evolution of kidneys starting all the way back in our platyhelminthes, our flatworms, where we talked about protonephridia. And so now we're starting to actually get to the function of what we're familiar with is kidney. And remember, the function of the kidney is just to regulate water, salt, and ammonia balance, as well as a couple other ions and things like that. But it's got to be drastically different depending on how I am or where I live. So I ask this question here: how does the act of peeing differ in a marine environment, so saltwater versus freshwater? So think about that. If I live completely in a saltwater environment, what is my pee like? Is it diluted or is it very concentrated? And likewise for freshwater. So just some funny little memes here. Um, to try to, maybe you have to stop the video here and go pee yourself, right? So let's look at a couple of terms here. You got osmoconformers and osmoregulators. So we're very familiar with osmotic. Osmotic means, again, relating to water. So what's the difference in the two? We got conformers and regulators. So if conform, I'm going to conform to my environment. Wherever I'm at, I'm going to conform in it. So they do not regulate they're just going to so if I'm in a salt water, my urine is going to be more. Well, we'll figure it out. I don't want to give it away just yet. And then you have again these osmoregulators, which have to expend that energy to control their water uptake and loss. Right. So osmoregulators, there's a lot of metabolic energy expended in regulating salt and water balance. Osmoconformers, very little energy spent. They're just going to depending on what the outside surroundings are, they're just going to be able to conform to that. They're not really going to regulate at all. So the example of osmoconformers are very, very early fishes, these osmoconformers, and I actually like that, is that in that early fishes haven't developed a well enough functioning kidney to regulate where they're living. So this here is the only vertebrate that's isotonic in seawater. So remember, isotonic is equal concentration in and out. So again, it doesn't matter what, I can put this in the Dead Sea, for example, and this animal is going to conform to the saltiness of the Dead Sea. If I put this in the Atlantic Ocean, it's going to conform to the different one. So it's completely isotonic, equal concentrations of water and salts going in and out at the same time. Amazing. Osmoregulators, again, have to spend that energy. And so we get the adaptation, the evolution of our very first kidney. But we've been working on this throughout the lineages, again, going all the way back to flatworms. So we've seen this. We get a little bit better as we get along. And even in our fish lineages there, we didn't just automatically start out with, I got a kidney. So you see in the agnathans, the lamprey and hagfish, that we're conforming. We have kidney-like cells where we're able to, sense different salt and water balances. Now I want to regulate and maybe conserve water, which is going to allow me to live in the marine environment. But if I'm in fresh water, I've got to be able to dump it. So we go from salt. That allows us to go the transition from saltwater, big ocean, to freshwater, smaller oceans, where I can possibly be a big fish in a small pond. So I have two different fish here. I have one in a freshwater and one in a saltwater. So let's look and see how their urine is different. And yes, this is a great short answer type question. So in a freshwater system, let's think about this. Where is most of the water? Most of the fresh water is outside of the skin. And so water is going to be absorbed every time that I open my mouth and I drink. Water is going to go into my system. Water is also going to be absorbed through my skin. And so when I pee, or when this fish pees, it's going to be excreting very, very dilute urine, because we're going from high concentration of water to low concentration of water. So a lot of water inside, and so, or I should say, there's a high concentration of water inside, and I'm going to be moving there. In the saltwater environment, it's basically the opposite. So think about you when you eat country ham or gargled salt water, anything really, really salty. What happens to the cells at the cellular level when I have too much salt outside my body? Water is then going to move outside to the salt to try to balance that. And so every time this fish drinks salt water, it's essentially making its cells around it thirsty. And so in order to combat that, my kidneys have to be able to pull out all the water, or lots of water, and I'm going to pee very, very, very concentrated urine. So hopefully that makes sense to you. I'll kind of explain it just briefly again. So think about a saltwater fish, lots of salt water outside. Every time I drink, I'm getting very, very thirsty. But I only live in salt water. I'm not getting fresh water. So I have to be able to conserve my water that I do get from the water that I drink. And so as a consequence, I'm going to have very, very, very concentrated urine, allowing my body to save any water that I do get. In a freshwater fish, it's quite the opposite. I've got so much water, I don't have anything to do with it. If I retain too much water, I risk my red blood cells bursting, causing other problems. So I have to get rid of the water, and so my urine is going to be very dilute. So, um, so make sure that you understand how the body systems differ in saltwater and in freshwater excretory systems. So again, saltwater fish lose water and thus conserve body fluids by having highly concentrated urine, while freshwater fish gain water and thus they have too much water and have to dilute it. So again, that adaptation of a kidney cell is just incredibly, incredibly important. It really set up the tetrapods very nicely as we move away from, depending on water environments, how can I conserve what water I do take, right? And so this is just a internal fish anatomy here. You really want to know this structure here. I like this diagram. It's very clean and very nice. So again, find something that you're very familiar with and you work your way through it. So the gills are typically always easy to find. Pathway of digestion, so of course the mouth, go through the esophagus, which is there, to the stomach, to the intestines. We see a couple big digestive glands like the pancreas right behind the stomach there, the liver, the gallbladder, right? The kidneys on top of the swim bladder there, reproductive organ, so whether sperm or eggs, depending on there. So if you've ever fished, especially right now, being that we're in April, we're probably getting ready to spawn here within the next, 3 to 6 weeks here. So when you open a fish, you'll be able to definitely tell if it's male or female. So a lot of the bony fish, you can't tell externally male or female. Anal fin, spiny dorsal fin, soft dorsal fin, ray fin, caudal fin. There's that lateral line that was able to sense the movements in water that basically a body ear, the nerve cord, and the brain. So, and of course, our two-chamber heart down below. So, swim bladder is there. So again, here's the swim bladder there. So if you catch a big catfish, typically you'll be able to see that swim bladder very readily. It's quite big. It's actually quite thicker than you would think, which is kind of nice as well. So there's a bust, and then because of a bust, now the catfish can only be at the bottom of the ocean or bottom of the rivers. So how does pulling a fish from 40 or more feet of water affect its size? And so if you've ever gone deep sea fishing, sometimes you're fishing 80, 150, 200 foot of water. You get a bite and you're reeling that fish up really, really, really quickly. You get to the top, you get your red snapper or whatever that you may have pulled up, and you may see this swim bladder exposed. And so what happened? Why essentially did we have an overinflation of this swim bladder really quickly? So what's happening is if I want to decrease and go down, my muscles around my swim bladder are going to tighten, making my swim bladder small. But as I go toward the surface, my swim bladder muscles are going to relax, allowing my swim bladder to overinflate. And so what happens generally in deep sea fishing escapades is that this swim bladder is basically overinflated too quickly. My muscles relax too quickly as I'm quickly going from depths of 120 feet to on top of the surface there. And a lot of times what the crewmates will do is they'll actually, like I said, they'll puncture this swim bladder, small hole there. It'll like I said, it'll heal itself. But what it does is it allows the fish to go back down toward the ocean instead of sitting on top of the water like a sitting duck, because it takes a while for the swim bladder to, and then in that case there, the swim bladder to decrease, allowing the fish to go back down to where it's supposed to be. So just a couple of things before we are finishing here for the day. Just to remember there, different fish for reproduction. Oviparous again, or sharks there. They lay their eggs. Really, I guess our fishes and chickens are the best. Is they lay an egg and it's fertilized. Lay it, forget about it. Oviviviparous is some sharks there. They fertilize eggs, stays inside the mother. There's no placenta, and they're nourished by the egg yolk, and the baby sharks are born alive. Viviparous is what we're familiar with mammals, being able to have a placental connection. Most mammals, placental connection, and our young are born alive. So what this sets up is we're going to go from the superclass osteo and look specifically at the class Sarcopterygii, these low fin fishes, and just a couple more slides before we finish up. Is that this class used to be a lot more involved and around a lot more species diversity than it is today. So currently there's only 6 species of lungfish today. But again, the fossil record there shows lots and lots of fossils of the Triassic. And these fish, these lungfish, as the name implied, low-fin fish or lungfish, their gills have atrophied or decreased to such a degree that they're actually not even sufficient enough for gas exchange. So they can't even breathe with their gills. And so as a consequence, they have to breathe atmospheric oxygen. So what structure do you think they have to have highly developed in order for them to breathe atmospheric oxygen? Obviously, they have to have some type of lungs, and they actually have 2 lungs, and so a right and a left. And so that right lung or a left lung will allow them to breathe atmospheric oxygen there and really set up the evolution of tetrapods. So I remember this back in high school in 2006 when this discovery came out. My biology teacher and chemistry teacher, biology teacher in particular, was very, very excited about this. And so when this fossil came out, it really was the missing link in our understanding of how tetrapods went from water to land. And so this here is Tiktaalik. This fossil here's an artistic representation of what it may have looked like. And so we have, again, these pelvic fins here, pectoral fins, have been modified into a pelvic and pectoral girdle. And if you've ever seen the original Little Mermaid from Walt Disney, and so the scene where she's kind of pushing herself up on the rock there, looking at the land, singing, I forget what song it is. But anyway, she's sitting there. This is kind of what Tiktaalik, what we hypothesize Tiktaalik has done, is that these fins allow him to go into the shallows and then kind of do like a little micro push-up. It would kind of invert its fins, push up, and then breathe oxygen air. And as we talked about in our evolution series, why would it be beneficial to move from deep ocean or deep freshwater systems into shallower and shallower systems? And so one of the possibilities is to escape predation. And so since there are lots of big animals, big predators in the big ocean and in the big freshwater, it would be a plausible hypothesis is that if I'm able to get into the more shallow flats and escape predation, then I can survive and reproduce. And those that were better able at getting into the shallowers and breathing this oxygen atmosphere, best able to survive and pass on their genes, allowing the next major class of vertebrates, the amphibians, to arise. But that's for the next video. So again, just a reminder here for this last exam, a fairly decent exam here. We're going to look at the characteristics of arthropods, echinoderms, and vertebrates. The taxonomy, again, I can't stress that enough, gets a lot of students. So spend some time with that. Get your hot sheet out. Get your your taxonomy sheet. Start doing that. Learn your evolutionary advances in jaw mechanics, respiration, circulatory system excretion, which we talked about today, locomotion and reproduction. And then again, of course, know your anatomical structures as well. And so looking at your guiding notes and making sure that we've covered everything before finishing up there. And again, I have this posted on eLearning Canvas for you, so absolutely. So we have done a good job today. I look forward to seeing you guys soon, and look for the next video to drop within the next couple of days. So see you guys.